So I'm a little bit, or maybe quite an outsider. Indeed, I have been interested in cardiolipins and lipids for a long, long time. But Barth syndrome, that's something entirely new to me. I'm so thankful to Ryan for introducing to me the importance, and I understand how essential it is to make a critical breakthroughs in this field. So as the first speaker, let me begin with the general introduction to maybe lipids. So now if we, in today's world, we have this universe of omics and lipids, of course. They represent a huge and diversified group of molecules. And so phospholipids, glycolipids, neutral lipids, and they all have a very similar plan of their construction. But if you think about numbers, then in today's assessments, lipidomes may comprise from 10 to maybe 100,000 molecular species of lipids. And if you look, this compares only with 20 to 30,000 genes and 70,000 proteins. So it's a highly diversified group of molecules. How do we get to so many different molecular species of lipids? Sorry, I have to kind of play both cards. So if you this is a typical phospholipid molecule, a two-legged phospholipid, and if both legs are different, as you can see here, so then the number of molecular species will be in hundreds. So now if you have a different type of phospholipid with 4 legs, and all 4 are different, so with only 12 fatty acid residues that comprise these fatty acid tails, so you're talking about tens of thousands of species. And if you take 20 fatty acid residues, you are now talking about hundreds of thousands of molecular species of lipids and phospholipids, particularly of cardiolipins. So this is good for a very rich, very significant, and highly diversified signaling language. You have to add to this, however, also that phospholipids and cardiolipin in particular can be polyunsaturated, and then molecules of this type are prone to oxidation. And with the addition of oxygen-containing functional groups to polyunsaturated fatty acid chains, and there are several of them, hydroperoxy, epoxy, hydroxy, and oxo groups. So now you have even a much higher diversification of phospholipid molecules. So you're evidently talking much more than about tens of thousands of molecular species of phospholipids. So how do you deal with all these kind of universe and oceans of different molecules? How do you assess them? So the easiest, and lately it turned out one of the most powerful technologies, is mass spectrometry. Essentially what you do, you kind of weigh with very accurately, with high accuracy, these molecules, and then you can identify them by fragmentation and looking at their kind of breakdown products. So that's a typical kind of spectrum of human brain cardiolipins. And I put this number here and indicate that the contemporary mass spec orbit trap technologies, they allow accuracy of 3 decimal points. Why is it so important? Because with this huge diversification, you need simple and accurate identification of these molecules. And if the accuracy is high as shown, then you can resolve isobaric species right away. So now one of the later achievements in mass spectrometry is imaging of lipids and building the heat maps of lipids. So the way it's done is that you can do it on tissue sections, and you take a tissue section and cover it with a matrix, and then you scan and obtain the spectra by shining a laser beam. And then, as you will see here, so wherever on the tissue section you see a signal, you can plot it and build a heat map. And so if you go 1 by 1, pixel by pixel, then you can get distribution of them, and you can overlay and compare it with the tissue section and get a pretty much very good understanding of distribution of individual molecular species of different lipids or phospholipids on a tissue section. And you can do it for phosphatidylserine, sulfatides, gangliosides, and so on. So how about cardiolipins? So until recently, cardiolipins were not quite kind of prone to this type of analysis, but with few modifications that we have been able to perform. So now we can see also distribution of cardiolipins on tissue sections as well. And so you can, in this way, you can detect and see and build these heat maps for all species of cardiolipin that you can detect, and then you can find which of them are oxidizable, less oxidizable, with longer carbon chains, shorter chains, and so on. So overall, this gives you a very good understanding that cardiolipins are a remarkably diversified group of molecules. And so their distribution in some tissues is very, very unusual and rich also. Now, cardiolipins are very ancient phospholipids, but if you compare the composition, molecular speciations of cardiolipins in prokaryotes with that of eukaryotic cells, you'll see a huge difference. So the red ones is cardiolipins from E. coli, and the blue spectrum is cardiolipins, mass spec of cardiolipins from mouse brain. So you see that eukaryotic brain, in particular cardiolipins, they are more longer chain phospholipids. MZ stands for mass to charge. If charge is 1, so it's essentially molecular weight. And prokaryotic, they are shorter chain and more saturated. Now, of course, at this meeting, we are mostly, and that's how evolution of this change has been occurring, that so more unsaturated and elongated species of cardiolipin evolved. So, but at this meeting, of course, we're mostly interested in one particular type of cardiolipins, this 1448 tetralinoleal cardiolipin. So, why exactly about it? So now let me show to you these two schemas that I borrowed from a very recent review by our outstanding, I think, review of our chairman, Dr. Schlamme. So in cardiolipin is among only two phospholipids, essentially, which is synthesized in mitochondria. And then, in addition to this synthesis, primary synthesis, it undergoes also remodeling. It's a kind of sophisticated multi-stage process in which monolyser cardiolipin can be reacylated to form, in the end, this tetralinoleal cardiolipin. And Barth syndrome, as you all know and we all know, so that's the syndrome, and that's a state where this process is, one way or another, is interrupted. So I wanted to show to you that cardiolipin, of course, is remarkably important for very many functions in mitochondria. So first of all, cardiolipin is responsible for creation of this curvature. So this is molecular modeling, coarse-grained modeling of cardiolipin in lipid bilayer. And if you put cardiolipin asymmetrically distributed to one side of this bilayer, you see that the membrane immediately gets this curved shape. So now we also know that the major biochemical consequences of tafazine deficiency, there are 3 of them. So it's a decreased in the content of intact cardiolipin. It's an increased level of monolyser cardiolipin, and it's also altered composition of cardiolipin molecular species. So which of these 3 is really something that leads to mitochondrial impairments and damage? So now we know that not only tafazine, but also mutation of 2 more genes and their products. So this prohibitins and DNA JC19. So all 3 of these genes and their products, they may be responsible for changed cardiolipin content, decrease. So then increased levels of monolyser cardiolipin, and then impaired electron transport in mitochondria. And people now say, and I think Dr. who is here who will talk to us about this increased reactive oxygen species formation, then possibly mitophagy and subsequent biological consequences. So now, mitochondria are damaged, but what do we really know about mechanisms of this damage, and what do we know about quality control of these damaged mitochondria? So let me briefly talk to you about this. So I need to apologize because I have so many things that I wanted to share with you that some of my pieces will be very fragmentary, and you'll have to trust me and ask me in the question and answers part of my presentation. So one of the most prominent features of cardiolipin's presence is in mitochondria, its asymmetry. So now cardiolipin as an endosymbiont, so it's kind of has been positioned in eukaryotic cells in such a way so that cardiolipin originating from bacteria, and let me play a little bit of music for fun, because I think we got too serious. Wow. I'm not sure how much can you hear of this. So this is a song of symbionts. So anyway, as a result of this, cardiolipins, they are present only in the inner mitochondrial membrane, and so the outer mitochondrial membrane does not contain cardiolipin normally, or contains them in very low amounts. And so what is the consequence of this? So if you look at mitochondria the way it's shown here, so mitoplasm, inner mitochondrial membrane, cardiolipin synthase, the sites and instruments of synthesizing cardiolipins. So most of them are not here, not in the outer membrane, but in the inner mitochondrial membrane. And you can detect it by mass spectrometry. You can get very robust signals from the cardiolipins in the inner, but not from the outer mitochondrial membrane. So what does it mean? What it means is that we know examples of this with phosphatidylserine and other phospholipids, that if for some reason this asymmetry of cardiolipin distribution is lost and cardiolipin can get externalized to the surface of mitochondria, this may be a label, a sign that indicates that this is not a good mitochondria, that it's lost a very important cardiolipin asymmetry. So externalization of cardiolipin triggers mitochondrial signaling. What is this exactly signaling? So you can look at this and model it experimentally. You can take cells in this particular example, it's mouse lung epithelial cells, and treat them, expose them to a proteinophoric uncoupler, CCCP. So in control, you see that if you label mitochondria with green fluorescent protein and also use lysosome labeling, so that these two do not co-localize. But in cells pretreated with an uncoupler, you see a lot of co-localization. So many of these mitochondria get autophaged. So now you can also, if you look at this moment and detect asymmetry of cardiolipin in membranes, you will be able to see that in conditions that are inducive of mitophagy, the externalization of cardiolipin becomes very obvious, a very significant multifold increase of the content of externalized on the surface of mitochondria cardiolipins in different cell types, in neurons, primary neurons, in neuron cell cultures, in tumor cells such as HeLa cells, and so on. Seems like this is a very common and uniform phenomenon. So now, then, again, excuse me for the lack of details. The general schema looks like this, that in normal mitochondria, cardiolipin is hidden from the mitochondrial surface, and then in damaged mitochondria, cardiolipin gets externalized, and then this externalized cardiolipin is recognized by this microtubule-associated light chain 3 protein, which is the major instrument of autophageal process, and these bad mitochondria with decreased membrane potential, so they are dragged into lysosomes and undergo autophagy. All right, so if this mechanism works well, so of course we need to understand and what is the kind of tools and how is this asymmetry of cardiolipin distribution, how is it disturbed? So what happens? So I wish I knew the answers to this, but we have to appreciate that if cardiolipin is initially localized here in the inner leaflet of the inner mitochondrial membrane, there are essentially 3 stages, 3 steps how cardiolipin has to get to find itself on the surface of mitochondria. Step 1 is migration of cardiolipin from the inner leaflet to the outer leaflet of the inner membrane. Then there's a big journey, and cardiolipin has to somehow jump from the inner to the outer mitochondrial membrane. And finally, it has to get externalized to mitochondrial surface. So what do we know about it? Not really very much. So thanks to work that we have performed jointly with colleagues in France and Canada, so that we have identified at least one of the potential translocators of cardiolipin, which is shown here, nucleoside diphosphate kinase D, so that can grab cardiolipin from the inner membrane and rotating as a kind of rotary machine transfers it to the outer mitochondrial membrane. And if you can mutate or knock down this enzyme, so then this process can be inhibited and regulated. So, but essentially the most important part is for mitophagy is appearance of cardiolipin on mitochondrial surface. So why is it so important? Because again, this is a quality control. Bad mitochondria that cannot generate and maintain their membrane potential, that's how they get eliminated, and this is very important for cells' health. So now, but what if mitophagy fails and somehow the damage continues? So what is next? So the next is a process which is called apoptosis, that these cells can be completely eliminated. So you can expect that in mitochondria with dysregulated interactions of cardiolipin with major mitochondrial respiratory complexes, you will have what is called ROS production. What exactly ROS, and what do I have in mind? So if these are cardiolipin and these mitochondrial respiratory complexes, see 1, 2, 3, 4, so then you can see that cardiolipin is essentially present in all of them, and it's essential for their normal function. So in dysregulation, what can be formed that electron transport will be carrying electrons to oxygen, and then superoxide radicals will be formed, and their dismutation will produce hydrogen peroxide. This is a very likely thing that may be happening in Barth syndrome mitochondria. So does it happen? Amazingly, I was not able to find a single paper on this subject. So, but essentially, what is the process that we're talking about? Again, bad mitochondria will undergo mitophagy. And this is a good pro-survival pathway, and if it works efficiently, fine, you don't need anything else. But if the injury continues and bad mitochondria accumulate, so then mitophagy cannot help. So this cell has to go. And this cell goes when cardiolipin undergoes peroxidation. And let me switch now to this. What do we know? Is cardiolipin oxidized in Barth syndrome? My answer, at least, I don't know. And I was not able to find a single paper on this subject, single study. So what do we know then about lipid oxidation and cardiolipin oxidation in general? So now this brings me to the point of mechanisms of oxidative stress and comparing it with oxidative injury and damage. So this is a typical schema that you can find in many reviews and textbooks, so that oxygen can undergo this univalent reduction resulting in formation of superoxide radicals, hydrogen peroxide, and 3 extra electrons give hydroxyl radicals. So then this will oxidize lipids, and the product will be lipid radical, which immediately grabs molecular oxygen and forms a peroxyl radical. And now this is the major reaction of lipid oxidation. So this lipid peroxyl radical may attack the neighboring lipid molecule. And if this is effective, so then this process may develop as essentially a very, very quick and fast, almost a chain type of reaction. It's a random lipid peroxidation. What is the chemical law of this process and this reaction? It's very simple, so that the easiness and readiness with which a lipid peroxyl radical attack neighboring molecule of lipid depends on the number of double bonds in fatty acid chains of phospholipids. If there is no double bonds, only one double bond, it's a very difficult process and oxidation does not proceed. But for lipids with multiple double bonds, this is a very effective reaction, and oxidation rate increases dramatically with the increase of the number of double bonds in the oxidizable lipid. So essentially that's the law of chemical oxidation. If in biological system what works is just pure chemistry, then you would expect that very highly polyunsaturated lipids, most abundant, they will be undergoing oxidation. Is that true? Well, by using mass spectrometry, we very thoroughly investigated lipid oxidation in 4 conditions where random oxidation was highly expected. So that was action of ionizing radiation. It was inhalation exposure, in this case, to carbon nanotubes in the lung. It's traumatic brain injury, and it's hyperoxic lung injury. In all cases, we were perplexed. We saw very selective oxidation of cardiolipin. Even in cases when cardiolipin was not the major, of course it's not the most abundant phospholipid, but even when it was much less polyunsaturated than other much more abundant phospholipids, such as phosphatidylcholine, phosphatidylethanolamine. How does this happen? And so what are the consequences of this? So here's my example. So it's a traumatic brain injury, and you can look at brain cardiolipins, and the blue ones represent normal, non-oxidized cardiolipin species. And the red one is this cardiolipin ox. That's a small amount of oxidized species of cardiolipin in normal brain. So now, after traumatic brain injury, acute brain injury, you'll see that the amounts of cardiolipin, normal cardiolipin, decrease. But what is more amazing is that the amounts of oxidized cardiolipin increase dramatically. And you can see it here as a diagram. In normal brain, there are about 10 or so species of oxidized cardiolipin. But after brain trauma, you see the formation of about 150 new oxygenated species of cardiolipins. So what does it mean? And do we need really all these 150 as signals or some kind of cell death signals in acutely injured brain? I'm about to exhaust my time. Let me show you. I was so puzzled and amazed by this that at one moment I thought, how can one better understand what's going on? Let me play you a little bit another piece of music. Do you hear it? It struck me that if you look at the masses of cardiolipins, so that they're between, let's say, up to 2,000 Daltons. And if you look at the kind of frequencies of the music that we hear, so we're approximately in the same range. So, and particularly if you look at the piano, for instance. So I thought, why can't we sonify mass specs of cardiolipins and see whether there'll be any difference between normal cardiolipins and cardiolipins from injured brain. I'm not sure how much you remember the previous melody, but there was a remarkable difference. There is a remarkable difference. So I don't know how much does it help to understand what exactly this oxidized cardiolipin doing, but I need to tell you that this may be a very interesting approach to kind of better perception of what happens to cardiolipins. So I guess, Michael, I have to rush, and will you give me 5 minutes? Okay. So how do cardiolipins get oxidized, and why is it so selective? So the catalyst of this oxidation is, believe it or not, is intermembrane space mitochondrial protein cytochrome C. So normally you wouldn't expect this activity from cytochrome C because all 6 of its coordination bonds are preoccupied by either binding and interacting with heme or 2 distal ligands, methionine and histidine. But it changes very remarkably and very dramatically when cytochrome C encounters now externalized cardiolipin. What you see here is, again, a computer simulation of membrane containing cardiolipin in blue, and this is cytochrome C. So when they find each other, so they really have very high affinity, develop interaction. And cytochrome C forms a very unusual and very strong complex with cardiolipins. And I'll show you now very quickly that in this complex, so this is cardiolipin with 2 phosphate groups and lysines on cytochrome C surface. And in this complex, so it begins with this electrostatic interaction, and then one of the fatty acids of cardiolipin evidently has a more profound interaction with cytochrome C, but it results in weakening of this methionine 80 heme iron bond, allowing small molecules such as hydrogen peroxide to sneak in and to start catalyzing unusual oxidation reaction. So I need to mention to you that formation of this oxidized cardiolipin is a required step in apoptosis. And if this chain of events develops, that cytochrome C encounters cardiolipin, so they form a complex which becomes a peroxidase. And then this peroxidase will oxidize cardiolipin. That this is an essential step in apoptosis. So my question is, can cytochrome C be involved in cardiolipin oxidation in Barth syndrome? That's a kind of very important. But why do we ask this? Because if you attempt to write this central, in my view, equation of apoptosis, that cardiolipin in the presence of oxidizing equivalents, or ROS, hydrogen peroxide, if it interacts with cytochrome C and causes cardiolipin oxidation, this is essential equal to apoptosis. Well, then the consequences are very clear. Once harm has been done, even a fool understands it. This has been said by Homer. Actually, a different one. So, but now let me just tell you very briefly that if this is true, so then this new peroxidase activity of cytochrome C cardiolipin complexes can be regulated and manipulated. And so we used and developed several of these. Nitroxides, these are kind of very interesting stable radicals that can prevent formation of superoxide and hydrogen peroxide. And now I have to run. So you can make these nitroxides mitochondria targeted, and so you can protect animals and cells from oxidation. And so another way to do it is to also kind of build molecules that will regulate heme iron orientation and coordination in mitochondria. And so you can lock the active site of cytochrome C as a peroxidase and prevent it forever from becoming a peroxidase. And in this case, too, you can protect mitochondria and animals against irradiation damage, as shown here, even 24 hours after irradiation. So there's still significant protection. I guess I have to finish here, although I have a small other piece. I just wanted to tell you that that dealing with cardiolipins and their oxidation is not easy. So you can block, of course, any oxidation forever, but is it good or bad? And I think that I just wanted to show. Wow, this is. Free radicals, toxins that destroy the body and the brain. All right, sorry. So you cannot eradicate all free radicals. All right. So let me show. Yes, this is my lab, and that's the way I see there myself, and that's the way they see me. Actually.
Valerie, thank you very much for this.
Valerie, Grant Hatch from Manitoba. Very great presentation. One quick question. When you've looked at those 4 different models of the generation of the cardiolipin oxidative species, does any particular species stand out that you see generation? Because those tissues all have different molecular composition of cardiolipin as their native species.
This is an excellent question, and here's what I want to tell you, that if you have multiple species of cardiolipin, and let's say you have one where you will have arachidonic, docosahexaenoic, and linoleic acid. Surprisingly, it's linoleic acid that will be oxidized in these cardiolipins. If you will ask me why, I have no answer. But we see specificity of linoleate containing species of cardiolipin oxidation, for sure.